Hey, everybody. Thanks for joining us today on our Sporecyte episode 6 of our monthly webinar series. Appreciate you joining in. This will also be up on our website for others who can't join us live. Today, as you can see, we're having the deep dive into mold and fungi, kind of what's the real problem behind mold. So John Haines, or I guess I should say welcome to everybody. As far as questions go, ask questions as we're going through the presentation, put those in the chat channel, but we will answer all those questions at the end of the presentation. But as you're thinking of it, go ahead and put that in the chat channel. So a little bit about John. I've been working with John for about 4 years on this project. So not only is he a PhD from Oregon State, he's been the expert advisor for Sporecyte. So identifying mold spores, curating data sets, working with us every step of the way, how our reports look, what we provide. You know, John's an intricate part of Sporecyte and the TechSite organization. So he's been a senior scientist of mycology at the New York State Museum. He's also an adjunct professor from Union College and Russell Sage College. He's been an indoor air quality instructor at the McCrone Institute. So for those of you who might not know, which I certainly didn't know, is McCrone is kind of the preeminent, I don't know if we'll let John delve into that more, but it's for somebody who wants to be in this industry, be an analyst, that's where you go, is the McCrone Institute. So it really is the place to go in this industry. He's a member of the Pan American Aerobiology Certification Board, which you'll see the PAACB. That's what that is. So he's also been a teacher of numerous airborne mold workshops here in the US and elsewhere. I should say he used to hold a mold spore camp in upstate New York, and I never got to attend that, but our CEO was able to go to that and get to know John. So anyway, John's written numerous publications. He is a leading expert in this industry, and we are lucky to have him working with us at Sporecyte, and we're lucky to be working with him today and get this presentation. So with that, John, take it away.
All right. Well, thank you, Jim. And I want to thank Jim in particular and Dylan for getting me to do this. And a special thanks to Troy for shepherding me through the computer glitches that we've had. I am the token Luddite in a tech community. So here we go. Nature in harmony. Here's a forest that's thousands of years old in Washington State, and it works perfectly. It has trees that are hundreds of years old, and but they have a life. They have an end of life. A forester once told me that a healthy forest has 20% dead trees, and that's what you see in the foreground. That is being recycled into nutrients for the new trees, which you can see in the rear on the right-hand side. So the fungi are an integral part of that. And it's with the advent of human habitation, humans had a subsistence kind of existence here in the prairies where they had used fire to maintain those prairies. And that was a very sustainable situation also. Fungi keep the balance in the biosphere amongst all of the plants and animals. And we humans had figured out how to make these, we love our corn, make these giant fields from prairies to farms, but that to a sustainable system didn't look right to the fungi. So there are fungus diseases. This one isn't the major fungus disease, but it's one of the ugliest ones. This is the corn smut fungus, also called huitlacoche in Indian or Mexican if you want to eat it. And here is a picture of upstate New York, where I used to live. 200 years ago, this was a hemlock forest. And it was cut down by the early settlers to graze their sheep and raise some crops. And they built this church around 180 years ago. And they built it of wood on a stone foundation, used a few nails and hinges and some glass. In another century, it is going to be what we call a cellar hole, depression in the earth surrounded by those stones that are now the foundation. And the trees, the forest will be back higher than ever, better than ever. Now here is Chicago. We build these giant cities, but there's still fungus here. I've air sampled in Chicago, and there are fungus spores in the air. They are kind of waiting to bring this back into harmony. Now, fungi are most genetically related to animals, but they are ecologically most connected to plants. And here, many of them are very specific. Like this is the birch fungus, Piptoporus betulinus, and it's just eating away at this fallen tree. There will be nothing left of it as firewood in a very short season, very short time. Now, the plants are definitely the producer of the biological system through photosynthesis and sunlight and chlorophyll. They produce everything. The fungus recycles it so that it continue. Without the fungus, you would have sticks and leaves and just a big pile of sticks and leaves, and the whole system would go to would stop. So I mentioned that most fungi are tied to their host plants, and that determines what fungus is going to be in each area. It depends on what the plant cover is. There are plants like the tar spotted maple, only grows on maple. Here's a beautiful forest in the island country of Dominica. There is no overlap in the plants between that island and North America. Likewise, there's also no overlap in most of the fungi. Oh, the exceptions. The exceptions are those fungi that can just grow on petri plates, on sugars and starches, and also can grow on buildings, on sheetrock. And that's those fungi have, all fungi have a place in nature. They do not, they have a place in nature, so they don't need the sheetrock to grow on. They just find the sheetrock to be a very convenient place to grow. So now we have two categories of fungi: those that live on plants, on certain plants, and those that can grow anywhere and can also grow in our buildings. And it's the algorithms of Sporecyte and the analysts in laboratories to separate those two so we can see what's going on. Here's a kind of a dramatic slide. Nothing in this picture can grow in a house. Everyone is dependent on a plant. Most of these are wood rotting fungi, the spores from wood rotting fungi. A few, the longer ones, the Ascomyces, are probably from leaves or small stems. And you'll notice those little grenade-like spores, 3 or 4 or 5 or 6 of them there. That's Ganoderma. We're going to talk about that later. Now, here's another dramatic picture. These are all the bad actors that will grow in a house. This is a great picture. There is Stachybotrys, there's Chaetomium, there's Wallemia, there's Aspergillus and Penicillium. All of those are what we call indicator spores. They indicate that there is a water problem in the house. Now here, let's talk about the different parts of the fungus. There's the mycelium, which is composed of these little strands called hyphae. That is the body of the fungus, not the part you see. This is the part that's hidden. This grows inside of the substrate. And then there are the spores, which we know very well. The spores are for dispersal and for survival of bad periods. Then there is in between the spores and the mycelium is a way to disperse those spores and to grow those spores, produce them. And that's either the fruiting body, or let's go to the next. Here are the spores germinating into the hyphae, which will become the mycelium, the body of the fungus. This is a great picture. It shows those little snake-like hyphae going out. They grow at the exact tip. They do not expand when they grow. Think about it. These threads are growing inside of plant cells. They're growing in sand and soil. They can't expand. They have no room to expand. So they grow right at the tip. Furthermore, if they have proper nutrients, they can grow virtually forever. There's nothing to stop them except what stops them in our environments, which is competition with another fungus, running out of nutrient, or running into inhospitable conditions. So let's look at that a little closer. This is a diagram, and those yellow things are the food. That's the starch grains. Let's pretend that this isn't in the corn or whatever that has starch in it. And the red arrows going up are the enzymes being secreted out of the fungus into its substrate to degrade those starch grains into smaller components, the sugars, the simple sugars. And then those are back reabsorbed. And that's the digestive system of the fungus. It has a digestive system, but it's outside of the self. Here's another picture of the mycelium. You can see that it's highly connected. I think it's a good analogy, a subway for nutrients, and it is. All the nutrients then can travel through these little tunnels to the outermost part of the mycelium. And you don't always see this. This is a mycelium by an overturned log. And this isn't the individual hyphae, but this is a huge rope strands of many, many hyphae degrading that log. This is a good example. Here's competition. The black lines around those colonies are where it's meeting a competitor, and it's walling off with the little black lines. Each one of those lines encircles a different mycelium, sometimes of a different fungus or a different genetic part of the similar same species. You can see some little white pieces around that are actually fruiting bodies starting to develop. This next picture, I love this one. This is the marasmius fairy ring on a lawn not far from where I lived in New York. You can see it's greener in the middle. That is the mycelium growing at where you see the mushrooms, and it's releasing nutrients into the soil in the middle. So it's greener in the middle, and that tells you how far that mycelium has spread. Next year, it's going to be bigger. Every year it grows like tree rings. Now, if you were, or I would, to take all of those mushrooms away, we have done nothing to the mycelium. It's like picking the apples from an apple tree. The apple tree is going to be fine. In this case, the mycelium is going to be fine. It shows that now the mycelium is not analogous to the roots of the tree. It's analogous to the whole tree. And the mushrooms are like the apples of an apple tree, and the spores are like the seeds. So how does that relate? It relates to the growth on a house, on a sheetrock. If you remove the fruiting bodies, if you remove the parts you see, it still retains the part inside, the mycelium, which continues to grow and continues to produce products like toxins. Now, the spore is an amazing thing. Look at it. It looks so fragile. It looks so small. These are really pretty tiny. This is about 5 microns across, these little warty spores. So why don't we use a scanning electron microscope to look at them? We can. The preparation's a little harder, but mainly you lose the color, which we use a lot in determining what we're looking at, identifying. It also misses the internal components, particularly the septa, the cross walls. Now, this is a very ancient picture in the early days of transmission electron microscopy, but it's a wonderful picture of a spore. This is the same spore. This is another 5 micron aspergillus. And look at that wall. Look how thick that is. And it's composed of these different layers. 1, 2, 3, and then a black line inside. That's the membrane. It's a semi-permeable membrane. It is so important. It prevents the spore from drying out, being dehydrated and just shriveling up. But it also will allow water into the spore to germinate. So we see these big blobs in there, these 4 blobs. Those are not the nuclei. Those are storage bodies called vacuoles. The nuclei are these 2 little things down here, submicron in size. So that's what a spore is. Now, that spore is so resistant that there are studies that show that you can bring up ice from 50,000 years ago in the Greenland ice core, spread out the ice and find spores that will germinate. So if they've been frozen for a very, very long time, there are spores that will withstand hundreds of degrees up to boiling, sub-boiling temperatures and still survive. So anything that the human body can survive, most fungus spores can survive. So aspergillus in particular, or all the moles, have instead of a fruiting body like a mushroom, just have a stalk. And then at the head of the stalk are these spore producing cells. Now, aspergillus is a huge genus. We'll talk about that later. The fruiting body. Now that's your mushroom and so on. Let me get that up. So the mushroom of the fungus is produced at the end of the growing season of the mycelium. When the mycelium gets too cold or too dry, it will produce the mushrooms. So they grow. Most mushrooms come up in the fall. Most mushroom growth from the mycelium occurs in the spring and summer, all growing season. Furthermore, mushrooms have a very short life, like from 1 day to maybe 5, 6 days. And that has a lot to do with what spores are going to be in the air. Let's go to the next one. Here's a different kind of fruiting body, a different kind of mushroom. This puts on a new layer every year. This is a Fungi, and it produces massive numbers of spores. Let me just go through. We're not too interested in these mushrooms. Here's the Ganoderma that produces those little spores like this. And I just want to explain why that can't grow in a building. These spores, and it produces perhaps 1 billion spores in a season, or several billions, maybe 1 billion spores in a day, many million spores in a day. You can see it just flowing out in a orange brown powder. And those spores need to find a similar tree, a hardwood tree, invade that tree and grow into a large mycelium before it can produce that fruiting body, this fruiting body. And only then can it produce more spores. So from spore to spore may take 3 to 5 years or even more. And that's why it's not going to grow in a building. Now, the climate or the weather conditions have a lot to do with what spores are going to be in the air. This is, think about Tuscany in northern Italy, the grape orchards, grape vineyards, the olive trees, and the Curbularia, the Pythomaces, this ascospore, which I don't know, we just called him Mr. Wiggly. Those had a pigment to protect them from ultraviolet, because ultraviolet is very damaging to the DNA of the cell. It will denature it. So here's another lifestyle. On a rainy day or in the night when there's no sun are the clear spores. Now, next, we see how fast spores can change in the course of a day. This is a slit sampler that the slit moves through this slide at 2 millimeters per hour. So it starts at 8 a.m. Here is noon, 6 a.m., midnight, so on. And you can see that change in there. If you sample here, you get nothing. If you sample here, you get a lot. That can change in minutes. And that all has something to do with what your outside sample is going to be, your environmental sample that you compare with your building sample. So everybody's interested in what fungi can actually do to you. Allergens are the most common thing amongst fungi. That's the most common effect it can have on humans, and that's an individual relationship between a person, their immune system, and the numbers of spores and kinds of spores in the air. And also it triggers asthma, which is an underlying disease. Now, next are the respiratory diseases, the ones that can actually grow inside of your lung, right in your lung. Remember, the lung is not directly connected to your bloodstream. So it is still open to the atmosphere in a way. When you breathe in, you're breathing in spores of all kinds every day, all the time. Now, one of the diseases is aspergillosis, caused primarily by Aspergillus fumigatus. Fumigatus, now, okay, let me back up. There are hundreds of species of Aspergillus. Fumigatus is just one of them. They do many different things. Their last count I saw was 800 different species, a huge, huge thing. So Aspergillus, you're talking about a lot of fungi. And Aspergillus fumigatus is the one that is thermotolerant. It is also universal. It's everywhere. It's from the Arctic to the tropics. Chances are you are breathing a couple of fumigatus spores where you sit now. But since it grows in heated environments like compost facilities, it will be the one that survives and will be produced in massive numbers. And it's really massive numbers that you need to to grow in the lung. The reason not every fungus can grow in the lung is because most fungi don't tolerate body temperature, and they have to be of a respirable size, which is like 3 microns or less, to get all the way into the alveoli, the deepest part of the lung. So let's look at the next one, histoplasmosis. That is associated with bird and bat droppings. So where do you find those? Certainly in parts of the house where there may be pigeons or bat infestations. Those should be taken very seriously with a lot of respiratory protection, because you don't want to breathe Histoplasma capsulatum. And the last one that we're going to talk about, coccidioidomycosis, Coccidioides immitis, occurs in it occurs in the desert parts of California, Arizona, New Mexico, and on down in South America. I'm mentioning it because in the laboratory, it is the most infectious particulate. Now, if it grows in the soil in those areas, the people most likely to be infected would be the agricultural workers and the construction workers. Let me just say that none of these fungus diseases can be transmitted from human to human. They're all coming from the environment. Here's Aspergillus fumigatus, the way it grows. It has a distinctive head. The little spore-producing cells are kind of pointed upward, like a bad haircut. They are small. They average about 2.2 microns. That's really pretty small. Here's an epicoccum. It's probably 18, 20 microns or thereabouts. So these are very respirable size. And here's where they grow. Big commercial compost facilities, particularly the outdoor ones, are just emitting Aspergillus fumigatus. So if you work with that compost, you need respiratory protection. I'll just skip over these. Some of my former colleagues working on the compost, Ed on the right, Greg on the left. So originally, I was going to call this whole presentation, water is the problem, mold is just the symptom. And I really want you to take that in. Here's my favorite picture, probably taken by Terry Brennan some years ago, a house in upstate New York. It was a summer home redone from an old family farmhouse. The generation had ironically worked at the National Institutes of Health in Bethesda, Maryland, and used it as a summer cabin. And when they went away for the winter, they just left the heat on so the water didn't freeze up. And a hose on the dishwasher broke. And it was the hot side, the hot water. And it completely devastated the kitchen. But it had this effect of bringing this hot, warm air in and exciting all of those spores that were already on this ceiling. That's the takeaway here. These spores were already there. The water didn't bring them. They were there just like the spores are probably on the ceiling where you are. Look around. Do you have sheetrock? Most offices, most homes have sheetrock. And you can't see, but they are covered with spores that with the proper conditions will turn into this. The other takeaway, look at toward the bottom of the slide where the warm mist did not hit, there's no growth. Now, there's been a lot of protection from moving spores from one place to another. And I say, don't worry about it. If there's no moisture for them to germinate, they're just so much dust. Okay, maybe allergenic dust, but just so much dust. Without the water, they are not a problem. Spores are everywhere all of the time. So here's a rather poor picture of a piece of sheetrock or drywall or wallboard. So common in the building industry. The inside is gypsum, which is actually a calcium sulfate disulfide. It is not a good substrate for mold. Mold really doesn't grow on it, but it does hold moisture, and it holds the moisture to keep the paper on the outside. And it's the paper, which is made of cellulose fibers, that is the problem. Here's a very dramatic problem. You can see that the joint compound in between the sheetrock is not mold infested. Okay, let's get right down to it. Stachybotrys. Everybody wants to know about Stachybotrys, the black mold, the black toxic mold, the black killer mold. It is not a human pathogen. It cannot grow in the human body. It does not tolerate the temperatures within the human body, and it does not do well in overcoming the immune system. It just doesn't grow in there. So it is not a disease. It grows outside in woody plants in nature. It uses cellulose of grassland plants, little weedy plants. It uses that cellulose, and it finds the cellulose on the paper surface of sheetrock very inviting. It's actually a water-adapted fungus. It takes a lot of water to make it grow, and when the spores are first developed, they developed in a slime drop. So you can huff and puff on that, and the air won't disperse it. It's only after they're old and dried that they are dispersed by air. Now, it does contain a toxin, but it's just one of many, many molds that contain toxins. And it's a bad toxin, but it's not perhaps the worst situation. Now, toxins are taken into the human body by respiration, is the most efficient way. Next would be eating it, through ingesting it. And the least likely way to get toxin in the body is by touching it. And since it's really not going to be taken in by breathing in such quantity that the toxin will be damaging, it's really not the problem that you have heard about. It can be toxic to livestock if they eat the moldy hay, but we're not really going to eat that. And if it's wet enough to grow Stachybotrys, a situation is certainly wet enough to be accompanied by other molds, particularly by Aspergillus. It also has an odd thing that we know about and not very well investigated. It has kind of a fungus dandruff, little submicron scales on its fruiting bodies, and it also emits volatile organic compounds. Its reputation is far worse than its danger. So let's take a look at it. It's very distinctive. Its growth pattern of the conidiophores has these right angle branches, which are really quite odd looking. And at the tips of them are these spore producing bodies, which are very distinctive. And here's what the spores look like. And here you can kind of see that these were in little groupings. And before I mounted that in a aqueous solution, those would have been little slimy heads where they would not be dispersed in the air. I like this. This shows the different stages. The spores start out as being almost clear, as on the left-hand side. And then they develop, this is the way we usually see them, but then they develop further into this really dark side, dark phase. And the algorithms of Sporecyte has been able to distinguish all of these and put them together as Stachybotrys cardarum. Now, let me tell you about Stachybotrys. What we know, what we thought was just one species, cardarum, actually has at least 3 entities. One that has a satrotoxin G, I think it is, which is a pretty dangerous toxin. Then there is another one that has aflatoxins, similar to the ones found in Aspergillus, and a third strain or species or genetic group that has no toxins whatsoever. And I can't tell, I don't know who can tell the difference between those without doing genetic analysis. Now let's look at some air samples. And we'll see what the algorithm and what the laboratory person sees. Oh, this is a great slide. This is that one I showed you before. But let's do some paired slides, outside and inside. Here's an outside sample. I don't know the location of this. I don't know the season. I don't know anything about it. But I'm guessing it's in the late fall because there's basidiomyces spores. You see the Ganoderma spore there. And so it's in an area where there's some hardwoods. And that black thing on the right, I'm guessing, is a tire shred. So probably near a highway or a road. Now here's the indoor sample taken at the same time, the same location. Well, almost the same location. It's inside the house. And the blue things are skin cells, very conspicuous. And that shows me that this is in an inhabited space. And I don't see any problem here. I see some dirt. I see the little glassy, like broken glass pieces, irregular, shiny. Those are silica pieces. Those are the part of soil that is the clay. That makes up clay together with these organic bits. So this is just like dirt. Let's look at another pair. This one, similar, it has the Ganoderma outside. Inside, we see the skin cells again, and we see an Aspergillus. This one is just plain bad. It has a lot of organic material, and it also has some starch up here in the corner. But it's not a fungus problem. I don't see a mold in there. Now let's just go forward a few more and look at different problems, fungus problems that we can find. Blue mold or black mold. This is a mold that actually is probably in a lot of wood that is already there in the lumber, not doing any harm. It's just sitting as the mycelium. And as long as the lumber is dry, it is just disfiguring. But people don't like this. This particular slide is from a very historically important rowing shell. And it's supposed to be varnished in clear varnish on the outside. And we want to get rid of those black spots. So this is the one place I would say, this and in timber frame homes, where you really want to use a bleach. You can either use sodium hydroxide, which is bleach, or you can use any of the, you can use oxalic acid to get rid of that. Otherwise, there's no reason to treat the black mold, the blue mold that you find in the lumber. If it's going to be not exposed, but it is unsightly. The homeowner isn't going to like his very expensive house timber frame to have that mold. And here's what it looks like when it's actually mature. Another specific problem. This one's unique to the whiskey distillery industry. It's the only place that I know where this particular fungus grows. And it really wasn't, people didn't pay attention to it until maybe 15 years ago or so. Now, here's a really dangerous one. Hormoconus resinae is a relative of Cladosporium that grows on oil or diesel. You can imagine if that grows on kerosene used for jet fuel, that is a very, very serious problem. So that's why much of the fuel, particularly the kerosenes and the oils, have an antifungal component in them. It also grows, and I didn't put a slide in, on timber frame houses or log homes that have been treated with certain oil products. Like even boiled linseed oil is a really good substrate. So your boiled linseed oil looks really great. And then a few years later, it turns quite black. So I think I'm going to end it there and ask for some questions. So Jim, take it away.
Okay, first one is, what's the relationship between fungi and bacteria, and how can we best describe this relationship to our layman clients?
Well, for the most part, they are separate entities doing different things. Bacteria are mostly related to animal products, whereas fungi are mostly related to plant products. Let me just go back to something pretty. I don't know too much about the underground activities in a forest. Obviously, connection between the bacteria and the fungus. It all works together in a system. As how you would explain it to a client, well, they both can be there. And the bacteria are not going to be so easily determined by air samples. You're not going to see them. And I hope that helps some.
Okay. So next, I'm not sure, let's see if this is a question or just a comment. It says, interesting info about Ganoderma spores. I've detected them inside a home of an old age and constructed of timber farming design in Greenwich, Connecticut. So I guess the question is, these finding Ganoderma spores indoors, is it likely coming from inside or outside, or?
Definitely coming from outside. And I guess I didn't really dwell on that whatever air is outside is going to come inside. And the only difference is if it'll be just come in as 100% if the windows are wide open. But if you have a system that will screen them out, which will have filters, it will be less in the inside. But you will always have some outdoor spores in your home.
Okay, excellent. All right, next question. When we're trying to explain the relationship between sheetrock and paper, the glue that encompasses the system, what is the best way to explain to a client that the drywall isn't the issue, but rather it's the paper and glue? And then next part of that question is, what is a good replacement for drywall? And when cleaning the contaminated area, what product is best? H2O2, bleach, or plain soap and water?
Well, let me do the one I really know the answer to, and that's plain soap and water. There's no reason to try to kill the spores and the fungus. You just need to remove it. And if it's at all more than just extremely minor, you really want to just replace the sheetrock. And you can't replace the paper of the sheetrock, as you know. I mean, you just have to replace the whole thing. And as far as the products, well, I don't know so much about that. But one thing I believe is that you do not have to kill it. And also, those spores that are so hard and resistant are going to be very hard to kill anyway.
Okay. Final question. At what point of exposure can we consider a building being diagnosed as having sick building syndrome? Do we need to have specific species at a certain load present, or is there something else we should be looking for? Good
point. No, you don't have to look for specific species. It doesn't matter whether it's Stachybotrys or Eucladium or Cladosporium spherospermum. If there's a fungus growth, that's something that people should not have to live with. So as far as how much is too much, there is no, I don't know of a good guideline there, but certainly there are guidelines about how many square feet are infected. And those you'll find those in protocols for remediation.
Okay, we got a couple more questions here. An inspector asked, the sickest clients I have all seem to have Stachybotrys in high numbers versus Aspergillus. Thoughts?
Well, I don't know. Okay, I hear you. So why are there such high numbers of Stachybotrys in air samples in a building? Maybe they only get there by being disturbed. Maybe maybe people are, once they see the Stachybotrys, which is so easy to recognize, they are perhaps not seeing or not paying attention to the much smaller, clear Aspergillus spores. Otherwise, I don't know how to account for that.
Yeah, that's a tough one.
Let me just say that so much about Stachybotrys is that it's feared, that there is a fear of Stachybotrys. Oh, it's not Stachybotrys, it's just Eucladium, or it's something else. We're fine. It's not fine. If there's a mold, you shouldn't have to live with it.
Okay. Let's see, we got a couple more. What are some common conditions or syndromes caused by mold or mycotoxin exposure that are regularly misdiagnosed? Can these be related to sick building syndrome, and are mycotoxins waterborne?
Let's have another webinar in the fall by somebody else. This is a little out of my area, and it's a really important issue, and I think maybe we can try to get somebody that can talk about that.
Yeah, I think that would be interesting, because I know as I talk to inspectors, and Dylan is always letting me know, like, we are not health experts and inspectors aren't health experts. So when you have the report, it's best to, provide that to the health expert who can work with the person to really identify what is going on, because we just don't know, what could be making somebody sick. And each person is individually, reacts to different kinds of things. So yeah, we just don't know. Here's another question. What types of mold are considered red flag or problematic?
For health, any of them. I would say any of them that are in large amounts in a building, whether it's an Aspergillus or a Penicillium or a Eucladium or even a Cladosporium. If there's a lot of them, that can be a health problem. It does not have to be Stachybotrys. Stachybotrys is the one that we have focused on, but it's perhaps not the only thing we should be focused on.
Right. And one thing to maybe think about too is, like, the Sporecyte report, we're really looking for using mold spores to identify moisture problems. So anytime that you would find a moisture problem, figure out how to fix that and then remediate, the site issues. For the health problems, use the report with a health professional for them to diagnose, but the report is not making the diagnosis. So another question is cross-contamination a risk in a limited infected area? So I'm not quite sure cross-contamination a risk in limited infected areas. I don't know.
Well, I think that goes back to that slide, the Terry Brennan slide of the really, really bad wall bar where there is the fungus only grew where there was moisture. So for if you have spores on your shoes and you walk through a building or you bring them home to your home, they're not going to grow. Trust me, those spores are already there before you walked in. And if there's no moisture, and it needs the moisture and the substrate, it's not going to be a house disease that is transmitted from one place to another, even from one room to another in a house.
Yeah, right. So for the mold to grow, it needs, the spores are all around us. So, moisture, environment that it likes to grow in, and then food for it to grow on, right? So
yes, exactly.
Okay.
And if there's moisture that continues, nothing is going to prevent it. In the long run, it's going to be back.
Yeah. Okay. Looks like we're getting another question coming across. So we have Beaver Valley in San Joaquin Valley.
Yeah.
When I take a report to a client, I tell them to get the report to their doctor and find a relationship between the two. Is this the best course of action? Should something test or urine test be the best indicator of exposure? Oh, blood. So should blood or a urine test be the best indicator of exposure?
Well, yeah, I'm not a real doctor. I'm a mycologist. So yeah, again, I don't know about the test. But one thing I do know, you're probably not going to be able to get the samples in an air sample. It's not about finding that. It's about testing the symptoms.
Okay. Another one, is the mold itself or the spores that cause? So is it the mold itself or the spores that cause the health issues?
Depends on whether it's a respiratory disease or an allergen. An allergen is just the spores. In a respiratory disease like histoplasmosis or aspergillosis, it's the actual fungus that's growing in there. And let me tell you a little more about aspergillosis. It's not always even noticeable. There have been cases where aspergillus nodules have been found in lungs of people that never knew they were there. These are, of course, not found routinely by cutting people open, but can be found in autopsy. And there was no evidence of respiratory difficulty because the little nodules were all walled off and the aspergillus was growing inside them.
Interesting. Okay, we've got another. We're getting this, continuous question. So that's really good. So the question has to do with airflow. Do you favor 75 liters over 150 liters? What are the benefits between the two? And I don't know. I think that's a question on sampling rate, and 75 liters is probably a total volume of air sampled versus.
Yeah, that'd be your 5-minute sample or 3-minute sample versus a 5-minute sample, or I'm not sure exactly. Can't remember exactly which. I guess you'll size up the situation. If you think there's a lot of stuff in there, give it a 3-minute sample. If you're a little skeptical and you don't see anything, smell anything, do a longer sample. But keep track of that the length of the sample that will give you the number of cubic meters sampled, or portions of a cubic meter.
Yeah, and I'll add one thing here. So Dylan could chime in too if he wants to, but we recommend for home inspectors that they do 5-minute samples at 15 liters per minute for a total sample volume of 75 liters.
Yeah.
So we find that in most instances in the home, the built environment that we're doing, that's about the right amount of for a sample. If you sample too long as particulates, hit the gel pad that's within the air cassette, if it gets overloaded, things aren't going to stick to it anymore if you get too much on there. Also, you just can provide problems for yourself. So unless you're in a very sterile environment, we find that the 5 minutes, at 15 liters per minute for a total of 75 liters is what we prefer. But if you do take something longer, make sure that you report that in the chain of custody, or the report would be inaccurate. So you need to say what your volume of air has been to reflect that. Our assumption is that it's generally, what we recommend, 5 minutes at 15 liters per minute. So if it's anything else, you need to note that. Go ahead, John.
Yeah, let me just say that the best mold detector is you. When you open the door of a building, from the first breaths you take, does it smell moldy? Does it not? So you're pretty much aware of a mold problem when you open the door, those first few breaths that you take. And you can use that to determine, are you really dealing with a mold problem in here? Maybe one that you don't see at first, but you can smell it. Some people, even with a lot of experience, can tell if it's a Penicillium, an Aspergillus, or a Stachybotrys.
Yeah,
I've been told.
I know there's dogs that sniff mold. I don't know if they can tell the difference, but I know there are some mold sniffing dogs.
I bet they are.
So let's see, we've got a couple more questions. Does humidity affect the samples? What happens to the gel pads if it's too hot or too cold or too old?
I don't know what the limits of that gel pad are. Certainly as it gets colder, if it gets really, really cold, it may have less retention. And hot, I don't think is going to be a problem, but cold might be.
I'll throw in a couple things here on this, and I think this would be a good FAQ for us that I could talk to the manufacturers about. But I do think if it's very cold, you want to let the gel pad, the cassette, heat up, warm up to room temperature. And Dylan can fact check me on this, because I think that gel pad could get a little too cold and things might not stick to it as well. I have done a couple tests where I, put the cassettes at different angles in a hot environment to see if that gel ran. And I haven't had it run, but I would say that, probably you would want to keep, a typical room temperature for the cassettes, like be aware of them. So don't put it in a hot car or a freezing car for long periods of time. As far as age goes, I do know that the manufacturers do put an age on those where they would age out. So as a lab, Sporecyte, we want to be really provide the best product for inspectors and for home buyers, building owners. So we do want to stay within the specified limits for ages on cassettes. So definitely be aware of what the expiration dates are on. It's usually written on the cassette. Just be aware of those. So another question, are expired cassettes not valid or not accurate? And I've never, in talking to manufacturers, they've never given me hey, this is a cutoff time, like, oh, you're good. It's like a food, best buy date. They have that expiration date. So I think our lab goes by, let's stay within the expiration dates. Let's not worry about, when the best by date is and when you can still eat that, can of peaches or whatever. It might not taste as good, but we want to stay within the specified limits of the manufacturers.
Well, let me just say that if you're going to end up in court to defend this, you definitely want to stay within the expiration date. As far as being out of expiration date, they probably work fine if you just want to see if it's there or not, if there's mold or not, and you're sure you're not going to end up in front of a lawyer as an expert witness or trying to prove something.
Yeah, that's a really good point. Okay, we've got a couple more questions. We'll take these 2 questions and then we'll end it, I think, because we're at 5 o'clock right now. So try and keep these within an hour. So these 2 more questions. Can we get a better idea of contamination from a physical sample? So I think that's a surface sample versus an air sample. Which is the best indicator of load or type of load?
Oh, I love tape samples or surface samples. Put it this way, that is the most direct thing. Of course, the most direct is, as the inspector, you're looking at the problem. You know how widespread it is. And the tape sample could be on a 1-inch spot, 1-inch colony, or on the entire wall, like some of those terrible situations we've seen. The sample is going to be the same. What the tape sample will tell you is it will pick up the actual mycelium. It will distinguish spores that are just sitting there from colonies that are actually growing. So I like tape samples. Air samples are one kind of filter away from that. They will just pick up the spores that are in the air at that time. Maybe the sample isn't being disturbed, or maybe it's been disturbed a lot, so that will affect the air sample. Now, live air sampling onto a Petri plate is one more removed, because it will not only just get the ones in the air, it will only get the ones that are viable.
Right. So,
but it has another, live samples do have another place in our sampling.
Right. It's interesting to note, like Sporecyte, we don't do any culturing of samples. In Europe, they do a lot of mold culturing samples. Mm-hmm. At least I've been told. So I haven't done any work over there. But as John says, for guys that are interested in cultures, the mold has to be able to grow on that medium that is in the Petri dish. So some molds won't grow in some of those Petri dish samples for that reason. So, but we don't do that. Let's see. Can samples, this is the final question. Can samples be taken in the rain under cover of an umbrella, or uncover, like, so let's say on a porch or under an umbrella?
Yeah. The sample may look a little bit different. If there's actually like a mist or a fog, the outdoor spores will cluster. They will gather together. Even though they're not maybe dissimilar spores, they will be involved in little little moisture drops and deposited on the sampler in the same way, on an odd little puddles of spores. But they'll be there. So you can sample. It just sample looks a little different.
Awesome. All right. With that, we will end the seminar for today, and thank everybody for attending and all the great questions. John, thank you for being with us today, and all the work that you have done and continue to do with Sporecyte. John's still an active member of our team and works with us all the time. So we'll end the webinar there, and thanks very much.
Okay.
All right.
End the webinar.
End the webinar.